Hello, welcome to Chapter 12. This will be Chapter 12, Lesson 1 for DNA. This will be the starting lesson for Chapter 12, Tolbert Biology. Think about it. How do genes work? To answer this question, the first thing that we need to do is to see what the genes were actually made of. Bacterial transformation. What clues did bacterial transformation yield about the gene? By observing bacterial transformation, Avery and other scientists discovered that the nucleic acid, DNA, stores and transmits genetic information from one generation of bacteria to the next. Bacterial transformation 2. To truly understand genetics, scientists realized they had to discover the chemical nature of the gene. This is one of the questions you have to answer in the Chapter 12, Lesson 1. So to truly understand genetics, scientists realized they need to discover the chemical nature of the gene. If the molecule that carries the genetic information could be identified, it might be possible to understand how genes control inherited characteristics of living things. The discovery of the chemical nature of the gene began in 1928 with the Danish scientist Frederick Griffith, who was trying to figure out how certain types of bacteria reproduce or produce pneumonia. And I know it starts with the P, but that's how you actually spell it. Pneumonia starts with the P. Griffith's experiments. Griffith isolated 2 strains of the same bacteria species. Both strains grew very well in the culture plates that he had in his lab, but only one of these 2 caused pneumonia. In the next coming slides, you'll see that I'll be talking about what's known as the S strain and the R strain. The S strain were the smooth strain, and the R strain were the rough strain. The S strain were the disease-causing bacteria, the ones that causes pneumonia, and the R strain, the rough ones, those ones were the harmless bacteria. Griffith's experiments 2. Disease-causing bacteria, S strain, smooth colonies grew on the culture plates, whereas the harmless bacteria, they grew rough edges. So that's what he noticed, and that's how he identified those 2 different types of bacteria. Remember, in the future, when you see the S, that stands for smooth, and when you see the R, that stands for rough. The smooth ones were the disease-causing ones. When Griffith injected these mice with the disease-causing bacteria, of course, these guys developed pneumonia and they died. When he injected the harmless bacteria, these mice stayed healthy. So he ran an experiment. Perhaps the S strain bacteria produced some kind of toxin that made the mice sick. He wasn't very sure. But to find out, he had to, he was a scientist. He ran an experiment. First, Griffith took a culture of the S strain and heated the cells to kill them. Then he injected those heat-killed bacteria back into the mice. The mice survived, suggesting that the cause of pneumonia was not some type of toxin caused by them. So he heated it up and killed them in the S strain. The lungs of these mice were filled with disease-causing bacteria. How could this happen if the S strain cells were dead? So this will be the end of lesson. This will be the end of lesson 1. There will be a general quiz that you'll have to take under Chapter 12, Lesson 1. Hope you enjoyed your time today. I'll see you under lesson number 2.